Welcome back. In the previous segment, we described the NGS technology that is used to obtain reads, which are multiple copies of short nucleotide sequences obtained from the genome. As we mentioned, these sequences are stored in FASTQ files. Typically, we obtain hundreds of millions of reads in a single experiment. Each one is of the length of 75 to 250 nucleotides, depending on the specific technology. In this segment, we will talk about the analysis. In other words, how we use the reads to find the genomic information of interest. Since the reference sequence of the human genome is already known, the first step in the analysis is aligning the reads to the genome, namely finding the location on the genome represented by each read. In principle, we could use the sequence alignment procedures we discussed in the previous units. However, given the fact that we need to align hundreds of millions of reads to a genome of 3 billion nucleotides, even use of the indexing algorithm implemented by BLAST will not be sufficient to handle data of this scale. Despite the fast computers available today, such an alignment could take days for each genome. The breakthrough that enabled the widespread use of NGS was not in computing power, but rather in the algorithms that allow the rapid search over millions of reads. We will not go into the details of the algorithm, other than to mention that it is based on pre-processing the genome and storing it in a data structure called suffix tree, which allows extremely rapid searches. This approach is called the Burrows-Wheeler transform, and by now it has many variants. It is an ingenious implementation of a theoretical algorithm from computer science that did not have much use to the most practical task in computational biology today, short read sequence alignment, and it works wonderfully. Following this computational process, we can look at the way the reads are mapped onto the genome using viewers such as the IGV viewer, although the subsequent analysis is generally done automatically by various software packages. The next step is known as variant calling, where we look for variants that differ from the reference genome. Since we usually obtain our fragments from both chromosomes, if we observed almost 100% of the same nucleotide in a given position, but this nucleotide is different from that in the reference genome, this indicates that the individual is homozygotic for this variation. Similarly, if we see that about 50% of the reads contain the reference nucleotide in a certain position, but about 50% show another nucleotide at this position, we know that the DNA is heterozygotic for a mutation at this position. So far, we have discussed DNA sequencing, but as we mentioned in the previous segment, the technology also enables sequencing of RNA molecules, a technology that is called RNA-seq. The analysis of RNA-seq is quite similar to the analysis we described for DNA sequencing. However, since we are dealing with quantitative results, we should remember that the number of reads that we see originating from each gene must be normalized. First, we have to normalize to the length of the gene, as longer genes will obviously produce more reads. We also need to normalize to the total number of reads in the experiment, which is often limited by technical issues. After normalization, the abundance of each gene can be stated in units of RPKM, which stands for reads per kilobytes of transcript per million mapped reads. This normalization enables us to compare the abundance of different genes and between different experimental conditions. Another complication we have to address is that mature messenger RNA transcripts are often a result of splicing, joining exons after ejections of introns. Thus, a continuous RNA read may be derived from two different exons which are not continuous on the original DNA sequence. Special variants of the alignment procedure were designed to address this challenge. In fact, These methodologies also enable us to study alternative splicing, a process in which the same gene can, under certain conditions, produce more than one, and often several, different transcripts that may lead to different proteins. Why do we need to do RNA-seq? The DNA of individual is similar in all of his or her cells, with a notable exception of cancer cells, which we will discuss in one of the next units. Thus, if we wish to know a person's genomic sequence, we would need to sequence their genome once in their lifetime. However, the transcriptome, the ensemble of messenger RNA that each cell transcribes, is dynamic. It can vary dramatically between each cell type and even for the same cell type under different conditions, such as stress, disease, or upon aging. Thus, we can gain many biological and medical insights by doing RNA-seq under different conditions and measuring the abundance of the transcription from each gene. Since messenger RNA transcripts are translated to proteins, the abundance of a transcript is insignificant, although far from perfect, correlation with the abundance of the resulting proteins. As we all know, determining the level of proteins in the blood is a major diagnostic and prognostic tool for many human diseases. However, standard clinical labs using biochemical assays can determine the abundance of only a few hundred human proteins. Thus, it became more clinically relevant to use transcript abundance as a reasonable proxy for protein abundance both in research and in clinical setting. The last step of the analysis of RNA-seq is to produce what we call heat maps, showing differential gene expression. In the figure, we see a list of genes that were identified as upregulated in the brain of older persons compared with younger individuals. This slide summarizes a meta-analysis, and each column represents a study that was done on many people. We can see that for some genes, in red, there is an upregulation of expression in older people compared to the lower level of expression in blue for younger people. Such studies could enable us to identify and subsequently to understand the roles of specific genes in aging. Another example can be seen in the next figure, showing the expression level of proteins in a person who, as a part of a research project, provided a blood sample for RNA-seq every few days over about 2 years. We can see the effect of viral infections on his immune system. The x-axis indicates days along the experiment, while the y-axis shows expression of several dozen cytokines, effector molecules of the immune system. Red color indicates, after normalization, that a specific gene was highly expressed on a given day, and the green color shows that expression of this gene was reduced. We can see a short immunological response after infection with rhinovirus that subsides after a few days, and much larger and longer response after encounter with RSV, a respiratory virus. This response lasted for about 3 weeks, and as suggested in this paper, triggered onset of type 2 diabetes. While we talked here about the response of the system in a broad term, it is clear that a detailed analysis of such maps can lead to much deeper understanding of the mechanism of the immunological response. These were just 2 examples of this technology, but it is clear to me that DNA and RNA sequencing will have a major impact going forward on the practice of medicine. In the next segment, we will describe more innovative and less obvious application of this technology. So please join us again.